In the last video, you have learnt about the different modes of asexual reproduction, that is binary fission, multiple fission, fragmentation, budding, spore formation and regeneration. Now today we will discuss about the next mode of asexual reproduction, that is vegetative propagation. It occurs only in plants. Vegetative propagation is the asexual method of reproduction in which new plants are produced or formed from the vegetative part like root, stem and leaves of old plant under favorable conditions. This process takes place in those plants which cannot produce seeds or which are failed to germinate seeds. Vegetative propagation is of two types, natural and artificial. So let us see about the natural vegetative propagation. It takes place naturally by the stems, roots and leaves of the plant. Now let us see the vegetative propagation by stems. The first is by the underground stems. The underground stems are modified for storage of food and that have buds. So these buds give rise to new plant. So in this method, new plants are produced from modified stems like tuber, bulb, culm and rhizome. So what is the example of tuber? The common example of tuber is potato. You must have seen some marks on the potatoes, and that marks are known as eyes. So these eyes on potatoes that you have seen are actually buds. So under favorable conditions, these buds develops into a new plant. Next is bulbs. The bulb is a small, round, swollen underground stem that have buds surrounded by fleshy scale leaf. So these buds produces new plants. Examples of bulb is onion, garlic. These are the common examples of bulbs. Next is corms. The example of corm is colocasia. So they have circular nodes. Colocasia are also the underground stem and they have circular nodes, and that nodes are unbranched. So from these nodes new plants develops. The next is rhizomes. Rhizomes are underground branched stems that grows horizontally. They have nodes and internodes and axillary buds. The new branches develops from these buds which later separates to form new plant. Rhizome stores food for unfavorable conditions. The common example of rhizomes is ginger and turmeric. Next is vegetative propagation by subaerial stems. In some plants, a stem grows or runs on the surface of the ground horizontally. So when the buds and root hairs of these stems drop into the soil, it gives rise to independent plant. Some examples of these type of stems is grass, mint, strawberry, etc. Next is vegetative propagation by roots. So plants like sweet potato and dahlia give rise to new plants from their fleshy roots which have buds. The next vegetative propagation by natural method is propagation by leaves. In some plants like bryophyllum and begonia, they have leaf buds on the edges of the leaves. When these leaf buds fall on the soil, they grow into a new plant. So these are the ways of vegetative propagation which occurs naturally. Now let us discuss about the artificial methods of vegetative propagation. Artificial methods includes cutting, grafting, layering and tissue culture. The first method is cutting. In this method, vegetative part of the plant that have buds on them cut, and the cut end is planted in the moist soil. So after some time, this cutting which have buds develops roots and grows into a new plant. You can take the buds of potato and leaf buds of bryophyllum for cutting. Mango, sugarcane, rose are the examples of some plants that can be propagated by this method of cutting. The next method is layering. In this process, the lower branch of the plant is bent towards the ground and covered with the moist soil after removing the bark of the part that is covered. So after some days, the roots are developed. The branch is cut and allowed to grow at new place. Examples of this type of propagation are jasmine, lemon, etc. The next method of vegetative propagation by artificial means is grafting. In this method, the branch of desired variety of a plant, desired which you want to grow. So the branch of a desired variety of a plant, that is called scion, is grafted or attached with the plant of same type having well developed root system, and that part is called stalk, which have well developed root system. It is known as a stalk. So after some days, you will observe that the tissues of the stalk and scion join to form a new plant. It can be seen in mango. Note this point that the cut stem which is attached to the rooted plant, that cut stem which is attached to the rooted plant is called scion, and the rooted plant to which the scion is attached is called stalk. The stalk is the lower portion and scion is the upper portion of the grafted plant. Now the next method is tissue culture. In this method also only one parent is required, and it is done in laboratories, or it is practiced in laboratories. It is a technique of growing plant cells or plant tissues outside the parent organism in nutrient medium in a germ-free environment. Now let us see the steps that are followed to perform this technique in the laboratories. First of all, select an explant. What is explant? The plant part, that is tissue or cell of the plant, which is taken from the parent plant, is called the explant, or the plant part taken out to be grown in a special nutrient medium is called explant. So select an explant. Now sterilize the explant. Sterilize means make it germ free. So sterilize the explant by using some chemicals. Why these explants are sterilized? They are sterilized to remove the unwanted microbes from the explant, because if the microbes are present in these explants, then when they get the nutrient medium, they also starts growing and make the hindrance in the growth of explant. So it is important to sterilize the explant. Now take a petri dish and sterilize it also. Then put the nutrient media in it. The nutrient media consists of sugar, inorganic salts, vitamins, amino acids, and some hormones. And this nutrient media gives the nourishment to the growing cell. Now put the explant in nutrient media. In the nutrient media, the cell or tissue, which is the explant, start growing, means it starts dividing. After some time, it will make a group or a mass of undifferentiated cell. Undifferentiated cell means that is not divided into stems, roots and leaves. So it makes a mass of undifferentiated cell. This mass of undifferentiated cells is called callus. Now treat this callus with plant growth regulators, means plant hormones like auxin and cytokinin. Auxin, that is plant hormone, it promotes the growth of the root and cytokinin, that is also a plant hormone. It promotes the growth of the shoot of the plant. Now when these plant hormones are provided to the callus, it promotes differentiation. These plant hormones promotes differentiation, means it forms the plantlets, which are the differential growth of roots and shoots. After 4 to 6 weeks, these plantlets are transferred to the soil in a pot, and finally these new plants are transplanted in the gardens or in the fields. So the advantage of tissue culture is that a large number of disease-free and high-quality plants can be grown in a short period of time. Now let us see the advantages of vegetative propagation. The first advantage is it is a cheaper, easier and rapid mode of propagation. The second is plants can bear flowers, fruits earlier than those produced from seeds. So it can grow earlier in compare of those plants which are developed from the seeds. Now the next advantage is vegetative propagation is the only possible method of reproduction for such plants that cannot produce viable seeds. Viable seed means seeds that can germinate. So it is the only possible method for those plants that cannot produce viable seeds, like plants like banana, grape, ginger. So these plants cannot produce viable seeds. For these types of plants, this method is the only possible way to propagate. The next advantage is parental features or characteristic of parent plant is retained in offspring plant. So the parent and offsprings are identical in all the characters that they have. So these are the advantages of vegetative propagation. Now let's see the disadvantages of vegetative propagation. The first disadvantage is new variations or the variations are not formed in vegetative propagation because the parent plant and the offspring both are identical in the qualities and characteristics. So no variations can be seen in this type of reproduction. The second disadvantage is it leads to overcrowding, means it allows a plant to produce a large number of offspring in a short period of time. So when a lot of plants develops in a very small area of the soil, then they cannot get proper or sufficient nutrient and light for their growth. So this is all about asexual mode of reproduction.